Good days, is Professor Will, CBMD PhD. Today's Wednesday, June 30th, 2021. It is 4:02 and 13 seconds, p.m. Eastern Standard Time. Today's research, Langerhans cell histocytosis. And don't forget to check out my website. So let's hop to it here. A promising two-punch therapy for Langerhans cell histocytosis. Date June 29, 2021. Source, Baylor College of Medicine. A novel combination therapy resulted in a significant decrease of disease burden in an animal model of LCH. Langerhans cell histocytosis, LCH, is a rare cancer involving dendritic cells, a type of white blood cell that usually helps defend against infections. The current standard of care of LCH, chemotherapy cures fewer than half of patients. The research team focuses on identifying the cause of LCH so that we can develop better therapies for patients. Most cells in LCH lesions are not abnormal dendritic cells, but other invading immune cells, such as T cells, that are recruited to sites of disease. The contribution of T cells and other immune cells to LCH disease is not known. The research histocytosis program characterized the type of immune cells in LCH lesions and the activity of those cells. Researchers wanted to understand the composition of the immune microenvironment in LCH and why the accessory immune cells are present. Are they trying to help and can't, or are they influenced by diseased dendritic cells to aggravate the situation? And there are two types of broad categories of T cells, one that battles and kills, called cytotoxic T cells, and another that assists in keeping the immune system in check, called regulatory T cells. Researchers found that both cytotoxic and regulatory T cells are present in increased numbers in LCH lesions. However, the cytotoxic T cells are paralyzed, a state called exhaustion, and are ineffective in preventing LCH lesion formation. Exhausted T cells are usually associated with a viral infection. After the infection is cleared, the immune system tells the T cells to stop fighting and retreat. As if raising a white flag of surrender, the T cells show a protein on their surface signaling that they are done fighting and are exhausted. In LCH, the disease cells trick the T cells into entering the exhausted state by triggering programmed cell death protein. 1PD-1 receptors, an immune checkpoint protein that turns the T cells off, preventing an attack on the cancer cells. And these exhausted T cells are not permanently damaged. If we can use therapies to turn off PD-1 receptor, these T cells can overcome the exhaustion and the associated functional incapacitation. Early studies treating LCH patients with drugs that block a growth pathway, an LCH MAPK pathway, have demonstrated rapid improvement in LCH lesions. However, responses may be incomplete, and lesions almost always grow back when treatment is stopped. In the current study, researchers described a new therapeutic option for LHC directed at the inflammatory immune compartment. The new approach uses immunotherapy to target the PD-1 receptors that incapacitate T cells, in combination with MAPK inhibition to target the dendritic cells that cause the disease. They hypothesized that the therapeutic combination would be more effective than either therapy on its own. The researchers measured the response to MAPK inhibitor treatment alone, anti-PD-1 treatment alone, and the combination of the two treatments. In models with the MAPK inhibitor treatment, the team observed a reduction in diseased LCH cells, but not infiltrating T cells. In models with the anti-PD-1 treatment, the team observed the opposite, a reduction in infiltrating T cells, but not the diseased LCH cells. But in the model using a combined therapy, a research group saw a significant decrease in both diseased LCH cells and infiltrating T cells. Researchers demonstrated that the combination therapy resulted in synergistic response with a significant decrease of disease burden when compared to either monotherapy. The results are very encouraging and highlight combined MAPK and checkpoint inhibitors as a potential therapeutic strategy in future preclinical and clinical trials for LHC. According to researchers, clinical trials testing combination therapy can offer hope to patients whose disease has relapsed. The combination therapy could also help reduce toxicity seen with the current standard of care. Good days is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.